Good day, dear students. I am happy to see you all in class. The year is still relatively new, and as such, I wish you all a happy and a very productive year ahead. That said, I am Professor Iyawe Hansen. By the grace of God, I and another colleague will be taking you the course BCH 407 titled Biochemical Pharmacology. Biochemical pharmacology is a course that combines biochemistry and pharmacology purposely to focus on the effect of drugs on biological systems and their chemical interactions. It is a 3-unit course for which you can easily score an A. Before we indulge in the course proper, it will be decent for us to know the course content and how we are going about it. We shall have in this course 15 lectures, which each focus on a particular topic, as we have here displayed. For example, lecture 1 will consider background information and definition of drugs. Lecture 2 will consider chemistry, classifications, and nomenclature of drugs. 3 will look at molecular basis of chemotherapy. In lecture 4, we will discuss the nature of drugs and transport across membrane. Lecture 5 will consider drug receptors and distribution. In lecture 6, of course, it will be metabolism or biotransformation of drugs. Lecture 7 will dwell on naming, overall reaction, multiple form of electron transport system, that is ETS. Lecture 8 will also consider inhibitors and physiologic functions of the cytochrome P450. Lecture 9 is the mechanism of drug actions. Lecture 10, molecular basis of the pharmacological effects of drugs. 13, we will consider elimination of drugs. Lecture 12, we will look at the biochemical actions of selected drugs. In this case, we will pick opium, Indian hemp, heroin, and others, or cocaine inclusive. Lecture 13 will be on action of anti-malaria and antibiotics. 15 will look at development and evaluation of new drugs. Of course, last but not the least, lecture 15, we will look at Nigeria traditional medicinal plants in the management and therapy of common ailments such as malaria, sickle cell anemia, common cold, and hepatitis. Now let's consider a very critical aspect, which is class attendance and course assessment. Examination of the course will be 70 marks. Continuous assessment will be 30 marks. Continuous assessment will be done online, and together examination and continuous assessment will make up 100%. That is 70 for examination and 30 for continuous assessment. Now, importantly, class attendance will be taken, and it's mandatory that every student must score 70% class attendance to qualify for sitting for the examination, in addition to the payment of school fees. That is how it goes. Now, for this particular course, there is a ready reference. It is a text called An Introduction to Pharmaceutical Biochemistry. This text is available at the University Bookshop. It is available at AAU Bookshop. It is recommended. It is easy to read and very easy to comprehend. It will guide you in case you are lost. Now let's proceed to lecture 1. Title again, Background Information and Definition of Drugs. Now, from birth, living organisms are endowed with innate immunity. These immunities are mobilized during infection to overcome pathogens. Apart from microbial pathogens, there are also ionizing radiations that the immune system or proteins protect us from. Now, drug discovery is credited to prehistoric people or the early men. Why is this so? It is so because they, that is, the early men, observed the healing powers, were the first to observe the healing powers of plants and animal parts. They also were the first to observe that plants or animals could be used against impairment of normal body functions. Also, they were the first to observe that some plants could be poisonous while others are not. And so, because of this realization, they used the roots, bark of plants, and herbs as food. Not just that, they also use them as crude extracts for the treatment of diseases. Now, as scientific knowledge advanced, it became possible to extract, isolate the active principles of plants and animals and then purify them. Now, it was this practice that gave birth to what we called, or to what is today called, pharmacology. And the concept of pharmacology actually began in the 19th century. The first drug to be isolated with the advent of pharmacology was the opium. It was isolated from plants. The experimental study of drugs and its action was established as an academic discipline in the middle of the 19th century. Due to improvements in knowledge and technology, it is now convenient to handle those factors that causes impairment of normal state of health. What is our interest? Our interest is biochemical pharmacology, which is the study of bioactive agents that impact on biochemical pathways. These bioactive agents are generally called or referred to as drugs. Now, what is the definition of drug? A drug could be any substance, either of natural or synthetic origin, capable of inducing a desired effect when ingested or administered. A drug is any substance, either of natural or synthetic origin, capable of inducing a desired effect when ingested or administered. The next question is, are drugs safe? Are drugs safe? Most drugs are used for treatment or management of conditions. They have their desired as well as undesired side effects. The desired effect is called therapeutic, while the undesired is referred to as toxic or poisons. Now, a drug being toxic or therapeutic depends on how you use them. We must know that all drugs are poisonous. All drugs are poisonous. Now, the difference between therapeutic dose and toxic dose is the concentration of the active principle you are taking in. When you take 2 tablets or a tablet of aspirin or Panadol, you have your headache cured or managed, as the case may be. At the same time, if you take 20 tablets of paracetamol, you are bound to die. So even when the drug can be therapeutic, it can turn poisonous. So it depends on what you want to achieve, if curative or death. So we can say that drug safety is contextual. It depends on the context under which the drug is used. So most often, drug safety is therefore said to be subjective. I have a set of study questions here for you based on what have been explained right here for you to assess yourself. You can go through it. It will be worth the while. This is the end of lecture 1. I want to thank you very much for your time. We'll meet again in lecture 2. Thank you very much.